Good morning. Aloha kakahiaka. David Lassner, Prez, welcome. Gwen asked me to give about a 20-minute speech outlining the history of research computing at the University of Hawaiʻi, but I shortened it, so don't worry. But I will go back in time a little bit because this is a really happy day for me. So in 1989, I got asked to be UH's first interim director of information technology. And at the time, all of the technology was managed by 4 different operating units that reported to 3 different vice presidents. And research computing really meant SAS, SPSS, some Fortran coding, and most of the research units bought and operated their own research computing, typically mini computers of some kind, and we were starting to see workstations, mostly Suns, pop up. The first thing I was asked to do in the job was to develop a strategic plan for IT, and part of that included research computing, and that was adopted by the Board of Regents in 1992, and it was created by a group that, because all of the IT was decentralized, there was a group called the Council of Directors of Information Technology. And the report at that time, so this is just about 30 years ago, research computing is flourishing at the University of Hawaiʻi. There are now some 300 Unix workstations on the Manoa campus, mostly in research units. The overall computing capacity this represents dwarfs the total power of centralized systems available in the university. And at the same time, UH was the first institution to formally join the San Diego Supercomputer Consortium when it was created. However, the amount of supercomputer time used by UH has not grown substantially. The San Diego facility is used so heavily that competition for the resource is severe. Also, network bandwidth between Hawaii and the mainland limits researchers' ability to use that center to take advantage of supercomputing and visualization technologies in a modern fashion. I think we had a T1 line at that time. Other than that first step, the university has made little progress in moving seriously into the computing areas attracting most attention in research today: high performance parallel processing and visualization of data. So this strategic plan also recommended creating a CIO, Chief Information Officer, position. This was increasingly common in higher ed, but then not universal. So 1994, new president comes in, Ken Mortimer, picks up the strategic plan for IT, waves around the booklet and says, "I hate to see plans that nobody acts on. What do we have to do?" So he assigned all of information technology to a senior vice president, Ralph Hori'i, who had business, finance, operations. Within about a month, Ralph called me into his office and asked me to be the new chief information officer. And he handed me an article from a higher ed facilities magazine, because that's what he did. I had never seen it before, but on the cover was a picture of the new IT center at Brown University, and he tells me I should start planning something. Little did I know how long that would take. But in '94, we created ITS, kind of the organization that you know about today. 2000, we do another strategic plan for IT that talks about high-speed internet access and high-performance computing. And interesting, this is the perspective in 2000. While investment in network infrastructure to support research is obviously a matter of system-wide and campus responsibility, over the past 10 years, the need for centralized computational support for research has become much less clear. Relatively affordable workstations can handle the computational needs of most researchers. Modest-sized servers can accommodate many research groups, and national or international high-performance computing facilities, such as on Maui, are available to university researchers via the internet. Furthermore, the computational requirements of applications and disciplines are so diverse that the economies of scale weigh heavily in favor of large national and international facilities with specialized disciplinary support teams. Earlier thinking that every university should have its own supercomputer has been replaced by the belief that every researcher who has an intense computational requirement should have access to a high-performance computing facility. National and international initiatives have emerged that are attempting to construct a computational grid that interconnects high-performance computing and related resources to provide researchers with seamless access to the information technologies they require, regardless of location. So kind of a slowdown on the whole notion of getting some kind of supercomputer here. Coincidentally, 2001, I worked with the executive director of RCUH, and we won the Maui supercomputer contract from the University of New Mexico. But by then, the security requirements were pretty strict. And so those resources never really became widely available to lots of our students and faculty. And meanwhile, Keller Hall, which was the venerable home of all of our centralized computing resources, was pretty close to maxed out. But the RTRF from MHPCC really has helped fund much of the work. So 2004, big flood on the Manoa campus, Halloween. Water gets pretty close to inside Keller Hall and pretty dangerously close to our machine room. 2008, we get funding to design an IT center. 2011, we get the funding for the building construction, kind of cobbled together with the support of the president, and some of the executives came up with some creative financing ideas. Late 2011, Dr. Gwen Jacobs of Lariat fame joins UH for a sabbatical with us. She was kind of trying us out to see if UH and Hawaii would be a fit for her. Is that fair? Apparently we passed the test. So in 2013, Gwen joined us as a full-time executive as our first director of Cyberinfrastructure. So I think I asked her to figure out condo research computing because I was kind of into that. The new building was well on its way. Well, you'll see today we get the testament of what she accomplished. I did also ask her, I saw this very cool financial model for how to fund with one-time funding, lifetime storage of research data. She didn't really pick me up on that one, but it's still a cool idea if anybody's interested. I got a good paper on the economics of that. But by the time Gwen actually started, I was off to my new adventure. I started as pres, I think about 2 weeks after she started here, maybe 1 week. And we moved into the new IT center in late 2013. Sadly for me, I never got to move in. Garret does have my name on his office if you ever go visit him, as a fond memory. But I will say this, my letter of appointment as president, I have no payout and I have no job when I'm done being president. But what I asked for and the Board of Regents gave me was the non-compensated title of President Emeritus and an office in the IT Center far out of the hair of any future president. So I will leave the rest of this story to everybody. Thank you, Gwen, for all you got done here. Thank you all for sharing your research, and I'm going to hang out and hear what you've been up to. Aloha.
Thank you, David. Appreciate it. Good morning, everybody. Garret Yoshimi, Vice President for IT. I'm the person that followed the other guy into the CIO slot. Always appreciative to have a boss that knows more about the topic than I do. It's great to have him around for air cover, air support, as well as to help drive us forward in an absolutely wonderful manner. So let me pick up the story where David left off. And yes, I do have his name on the door. My statement is, I'm just borrowing the office. The office is his for whenever he wants it and whenever he decides he'd like to use it. So David, you're always welcome to come back and reclaim your office at any time you're ready. We also have reserved a special space for you with a great and a wonderful mountain view out of the corner of the 6th floor. So you're also, we've set aside the space for you to be able to figure out to do in your retirement years at some point in time, but don't do it yet. Hang around for a while. We want to keep you here and keep you engaged and help us to move the program forward. A couple of other things that I wanted to add to David's remarks. We had the fortunate experience of having a fairly large system investment placed in to an HPC cluster. This was the prior CIO, this wasn't me. He was able to both muster resources as well as support during the period where he was able to raise funds to not only build the building, but also equip it in a manner that is highly supportive of not only our role in the enterprise space for students, faculty, and staff, but also in the research space. So the initial investment was really key to bring the initial cluster online and up to speed. Our initial Cray cluster was probably brought online in about 2015-ish, 14? Okay, 14, yeah, 14, before I started, which really, speaks a lot to the commitment of resources to our researchers and in particular to our students to make this happen. In addition to the resource commitment, the condo model of being able to build and expand the HPC cluster is absolutely key to making sure that we have a sustainable operation on a going forward basis. As many of you know, the one-time money is really great, but the secret sauce and the key is really to be able to have a sustainable financial model on a going forward basis to make sure that those resources don't just simply age out in place and then at some point in time become just a space heater and not that useful to its original mission. So it is really about making sure we have a sustainable program that can also fuel the growth and the demand for the resources in this space. As you'll hear later this morning, a bunch of the different lightning talks, as well as the conversations of folks that are using and making use of the resources are absolutely key to the mission of the university. So we're happy to be able to support that on a core basis going forward. I wanted to mention one other aspect of the HPC cluster is that we actually use the free clusters or the free cycles that are available. And you know what, frankly, they're available on any computing cluster, but systemically use the free cycles to be able to deliver services into our researchers, especially the folks that are starting up in the field, as well as to our students to be able to use and make use of this class of resource that they would not otherwise be able to buy into, but be able to use the class resource, get up to speed on it, and then write their own grant proposals and program proposals to be able to grow their research as well as help us sustain our cluster. So let me thank everybody for attending. I see we have a lot of folks. I see Sean. I guess we couldn't get you on a plane, but we're happy to have you working full guns for us. I really appreciate your work in the space, and all of the other friends and folks on the screen that are really instrumental in keeping the program moving forward. I thank everybody again for your hard work and your efforts to keep us moving forward. And I will turn the floor back over to Gwen to keep us moving forward today. Thank you very much.
Thank you, sir. All right, and thank you, Michelle. Here we go. And I can just advance with the return, right? Good morning, everyone. First of all, I want to thank both David and Garrett. Oh, am I okay now? There we go. Okay, awesome. Still have a little bit of an echo. That's that. Great. As you can see, I have an incredible staff that works with me here and I wanted to, before I get started, first of all, to thank David and Garrett for their remarks. Have to say the best part of my job is working with these two guys and having their support over over this long journey that we've done to build cyber infrastructure here. And then secondly, I want to just have a shout out to everybody on my team. David Lassner back there is on my team, sorry. Ron Merrill, Sean Cleveland, David Schanzenbach, Jennifer Geis. Oops, where'd Adriana go? I don't see Maria and Michelle. So our team could not be as productive as we are without all of your good work. So thank you so much for everything you do. And I think all of the scientists and researchers that we work with know how great it is to be able to work with all of you. So what I want to do is just take a little bit of time to go over some of the history of the HPC, also talk a little bit about the kinds of users we have and some of our plans for the future. So this diagram shows basically a timeline for the investments in building the HPC. Back in 2014, David worked his magic and we were able to get 1.8 million to actually buy our very first supercomputer, the Cray. We opened for business with seven early adopters in 2015. And by 2016, through the condo model that both Garret and David have talked about, we then received another $733,000 in PI investment. And these are folks that are buying their own nodes on the cluster and contributing to both the growth of the cluster, but also the maintenance as well. Again, in 2017, so obviously our condo model works really quite well. Also, I think everyone knows this, that anyone can use the cluster for free. You don't have to buy a node to be able to use the computing resources. And I think some of the scientists that you hear from today will talk a little bit about how they've been able to use some of those free resources to do a lot of their great work. So in 2017, keep rolling on with our PI investments, almost another $300,000. And then we made another institutional investment in changing over some of the control systems of the cluster. And then again, another investment from our PIs. In 2020, we wrote an NSF MRI award, which we won. And that was primarily to change up the composition of the cluster to start adding more GPU nodes to the cluster, because this just reflects the changing nature of science, also recruiting a number of great researchers that use machine learning and artificial intelligence. And these kinds of processes are much better for that kind of work. Rolling forward, another investment in 2021 and again in 2022. And now we're crossing our fingers, but we know this is pretty certain. Just want to congratulate Sean Cleveland on winning a CC Star Award from the NSF. So this $400,000 will really add additional computing power to the cluster, but also start to build out a very high performance storage system. So as you might guess, the needs of our researchers have changed over time. And part of our goal here is to stay on top of the latest technologies and be able to have a resource that really reflects the needs of our community. So if you look, it's about $4.5 million total investment in HPC. UH has played a big role in that, as well as some of our grants and our users. So I think we're very pleased with this progress. Also, as I discussed earlier, we've been building the cluster over time. We started out with 38,000 cores and no GPUs, and 7 early adopters who kind of road tested our cluster in the beginning. And as of present, we have 726 total users. They don't use the cluster all the time. We have about 353 active users, but we've now expanded basically the CPU core of the machine out and also added a number, 120 GPUs. We'll add some additional GPUs and some more cores and more storage with the CC Star Award that Sean won. This is a little bit complicated. We'll have these slides for folks if they want to take a look at this afterwards. But what this slide does is it chronicles how we built up the cores and the memory and the various different types of processors that we put into the cluster over time. As I said, our goal is to reach out and be responsive to our users. We work with folks we have a faculty advisory committee that meets and gives us feedback on what kinds of purchases and things to make in order to keep our cluster both current in terms of technology and also large enough to be able to support our user base. We wanted to show you just, and I know this is a little bit hard to read some of the text on the side, but just to give you an idea of the breadth of users that use our cluster. So some institutions have an engineering department and they have a cluster and only engineers use that. That's not our model here. Our cluster is open to any and all disciplines. And you can see on the left, basically the number of users by discipline starts with biology, math, atmospheric science, oceanography, physics, geophysics, chemistry, and also social science. So we're really covering all of the bases here, we think, in terms of the research needs of our campus. If you want to look at the breakdown between what types of computing technology these different groups are using, physics and astronomy is using a lot of our CPU hours. I think you'll hear some of that from Philip later. And but this just gives you an idea of how this resource is used and how broadly it's used by researchers, not only here at UH Manoa, but across different campuses. And also, we're delighted to have Renee Bellinger here today when she gives her talk, one of our top users from UH Hilo. So this really is a system-wide resource, and we're happy to be able to support our researchers across different islands as well. Okay, publications. So on our Data Science Institute website, we have a list of all of the publications that from users who have used the cluster in their research. I think this is one of the most interesting ways to show just the breadth and the quality of the research that's coming out, not only from our scientists, but also that's enabled by our by our resource. So right now we have more than 130 papers. I think there's probably more. Not all of our users let us know, and we encourage them to let us know when the cluster has actually helped out on one of their papers. And we're delighted to say that many folks not only acknowledge this, they include a very generous acknowledgement of not only the use of the cluster, but also highlighting the help of many of our staff members. So that makes us feel good. We're glad to be able to do that. So if you're interested in kind of the breadth of research, you can take a look at that link and see all those different publications. Now it's my great pleasure to be able to start with the lightning talks. Today we have 6 different lightning talks. I think these are really indicative of the different ways in which our users use our cluster, and from a number of different science domains and also research individuals. So what we're going to do now is we will start with the lightning talks. Have about 7 minutes for your talk. We will take questions. We'll have a little moderated Q&A. So we have a reasonable crowd here. I'm sure you'll get some good questions. So I think we are set. Are we set, Michelle, to start with Philip?
Hello, can you hear me? Let me share my slides.
We can hear you.
All right, I hope you can see my slides. So, hello, good morning. So I'm very happy that I was asked to give my presentation here on all the work that I've done on our nice Mana or HPC system. So my name is Philip von Doetinchem. Most students call me Von D, so that's a bit simpler. And I'm an associate professor in the physics and astronomy department. So, and my group is called the Cosmic Ray Antimatter group. So I'm a particle physicist by training, but I conduct particle physics essentially in space. And one thing that we're really trying to investigate more is the existence of dark matter. So I do not want to go into the details what dark matter really is, but you have to imagine something like all the regular matter that we're made of, like protons, neutrons, electrons, all the stars, all the suns and planets that we know make up about like a few percent of all the energy that we have in the universe, and dark matter makes up at least 5 to 6 times more of what we kind of know. So this matter is called dark matter because it does not shine like regular stars. And what you see here on this picture is a collision of clusters of galaxies where you can really see like the different distributions of dark matter and regular matter. So by now we know dark matter exists. Okay, let's hold this thought for a moment here. And now this is more like the astro part of my work. So now how do we study dark matter is we're trying to use messengers similar to what you do with a regular telescope, where you look at photons coming from a star. We're trying to use charged particles, for example, something like protons or electrons that are produced in our galaxy, and they might tell us something about the nature of dark matter. So this is a complicated process. Obviously, we cannot just go to the origin of like wherever this matter is created, but they are created in some process somewhere, and they scatter around in the magnetic fields in our galaxy. Eventually, they interact with our solar wind in the solar system, and then they get closer to Earth, where we finally find our detectors, and they interact with the geomagnetic field, and then eventually interact with our atmosphere, where they create a big air shower. So understanding all of these different effects is a complicated endeavor, and I'm involved in a number of different aspects. So just to connect these two different topics that I introduced like way too briefly. So on the one hand, we have dark matter. On the other hand, we have cosmic rays that are produced in our galaxy from a large variety of processes. And now we try to understand dark matter better by looking for a very specific type of cosmic ray, which could be antimatter. So antimatter is like regular matter, but it just has the opposite charge. So for example, everybody knows a proton, hopefully, that makes up like the large fraction of the nuclei that we were made of, but like an antiproton is just the same as a proton, but it has a negative charge. All right, so the advantage of studying these types of particles is that there are very few sources in our galaxy that can produce these kind of signals, and by having the signal that is like, or the messengers that we're looking at, that is hopefully background free, like that there is no other process in the universe that can produce them, we're trying to look for signals in antimatter that then hopefully can tell us something about dark matter. So it's kind of a bit convoluted. And to do this, I'm involved in a number of different experimental efforts. So we have the currently operational system AMS-02 that is operating on the International Space Station. That will go a few more years until the ISS is decommissioned. I think that will be around 2030. And we're currently in the process of building a new balloon experiment called GAP that fly from Antarctica hopefully very soon. So that is coming along. And you also have to conduct measurements on ground with an experiment like NA61 SHINE, which is located here at CERN in Geneva. So you have to make all of these different measurements to make progress in this particular field. And now, talking a bit more concretely about what I used, or my group used the cluster for. So in 2015, I don't know if it qualifies as being like an early adopter, but as soon as it became available to like kind of the regular user, I was very interested in testing it out. And in 2015, I started out with simulations of interactions of charged particles in the geomagnetic field here. So you see this figure here on the left. I hope you can see my mouse here. And you see all of these lines correspond to charged particles, making these sometimes like very convoluted trajectories in our magnetic field. And we have to understand what these different trajectories are to understand if these, in this case, antiprotons came from outer space, or if they were created in the atmosphere, to really make sense out of the data that we're collecting with our experiments. So at this time, I was very impressed. Like in a very short amount of time, I could get hundreds of years of compute time out of the HPC. So that was really, really brilliant, and allowed to do some studies that were not possible before. So then one other study is kind of depicted with that little cartoon here in the middle. It's not doing it justice how complicated it actually is. And my grad student, Anirvan Shukla, used about thousands of years of compute time to have an event-by-event simulation for the creation of anti-heliums. Anti-helium can be created in proton-proton interaction, but it's extremely rare. It's extremely hard to produce, and we really needed this computing power. Nobody had ever done this type before, and some people said, oh, you can't do this, like you need way too much compute power for that, and we made it possible. So then on the far right, we see another aspect of what real particle physics is about. So we have particles interact with our detector, and then we have to make sense out of whatever we measured. And this can be like a complicated pattern recognition system. We call it the event reconstruction. And this is what we're heavily using for our new GWAS experiment. So for the simulation of these interactions and for the reconstruction of these events, we're using it a lot. So we use it for the design of the overall payload. We use it for the design of our algorithms that we will then use for the actual data that we will collect hopefully soon on the first balloon flight. And so this project is led by my postdoc Achim Stössel, and there are some other projects coming on. And so we receive much more help in other projects as well. So just to highlight a few things. Closing here, I would really like to conclude by saying that Mana enables cutting-edge, world-class research of far-reaching relevance. So what we do here as this group is not only benefiting our local group here, but particle physics is very national, international, and many of our partner institutions, MIT, Columbia, Berkeley, just to name a few national institutions, but also international institutions like the Italian Institute for Nuclear Physics or the Japanese Space Agency. These are all collaborators, and they directly use the products of what we produce for their research. So we also kind of enable really something that happens on the national, international scale. And I would really like to then finally close by expressing my gratitude for all the hard work of the whole Mana HPC team. So we have had lots of great interactions over the years, and you made our research very successful. I'm very grateful for that, and it's always very enjoyable and fruitful. So thanks a lot. I really appreciate it. Thanks a lot. That's it from my side.
All right, thank you, Philip. Any questions for Philip from the room or anywhere? Anybody have a question? Okay, with that, I will turn it over to
Okay, I hope everyone can. Okay, I hope everyone can hear me without an echo. All right. Thank you very much for inviting me to this exciting symposium. I know this is a 7 years celebration with Mana, but I'm only with Mana in the last 4.5 years, so I don't really consider myself as an early adopter, but I thank you guys. So my talk gonna be, it's only 7 minutes, so I will give a very brief introduction of my research that concerns around the star forming region chemistry and also how our cell works. Here are really 3 focus of my research. First is the reaction dynamics. We're interested in how does a chemical reaction take place. We also study machine learning to accelerate the simulations that we're doing. And the last is the biophysical process. We would consider ourselves as a membrane expert. So we simulate all sorts of membranes and understand what it does to facilitate a lot of bio functions. So here is the first topic, that is chemical reaction. We know that in the outer space, the molecules collide and they could form different kinds of product. Here is just an example. Very simple molecules, one come from left, one come from right, they would collide. They would form different sorts of product. However, this kind of reaction is very fast in the space, usually on the time scale of femtoseconds. So it's very, very difficult for the experiments to measure. What we did is we developed a application molecular dynamics that, as you can see the animations here, also is a little bit lagging. The idea is we could follow the motions of atoms in real time, and we could really understand a lot of non-traditional behaviors. And also because of Mana, in addition to those research, I also worked on other projects, and sometimes you got some surprises. For example, these examples of media coverage is once upon a time, I stumbled on a computer program that is really well adopted in organic chemistry study.
And we realized this computer program published 3 years before I noticed there is a bug, has been used over 100 times. And we found this bug, we published the fix, and it attract international attention. The idea is, what do we do with open source computer programs that have glitch in it? And because of the importance of this research, our group is able to secure 2 fundings from Department of Defense. One is working on hypergolic ionic liquid. The other is working on to develop the next generation energetic materials. So those would be used in NASA space programs and also other space vehicles. These simulations that I just mentioned to you looks very great, but it's very expensive. The idea is you would have to compute the force involved in the chemical system using quantum chemistry softwares. And you have to propagate your system over and over again. So this is an iterative process. Because of that, we're working on to develop a machine learning protocol that will be utilizing the force or the ab initio gradient that we already got to propagate our new trajectories. And this is our fundamental importance to just in general, the molecular dynamic field. Therefore, it is awarded another NSF award just recently. Okay, so this is the last thrust of my research. Like I said before, I would like to consider myself as a kind of a membrane specialist. We do all sorts of membranes, including vesicles, drug permeations, and the tear films. Mana has been really able to provide a lot of computing power on this front. And as a result of that, we're able to form a collaboration with Eli Lilly that has been going on in the last 4 or 5 years. And we're able to study the drug permeation to predict the permeability of really any arbitrary small organic molecules. We also want to make our contribution to fight against COVID-19. This is a collaboration with a Professor Shikuma in JAPSOM. And what we're interested is we want to figure out whether some of the FDA already approved drug could be used to block the migration of COVID virus. And this report, and along with other report, was reported by the Wall Street Journal. And also because of the superb computing power that Mana is able to provide, we're able to simulate the first ever atomistic simulation of a full-scale vesicle. So this has been previously considered too large for all atom simulations, but we're able to do that with Mana. And as a result of that, we generate enough preliminary result to make it believable, feasible, so that we could get more computing time in Anton. This is a specially designed computer that is considered one of the fastest in the world, and we were able to get 7 days computing time on it. We're currently using it. So that brings me to the last slide. Here is, like I said, I joined UH Manoa roughly 4 years ago, and this is currently what my research group looks like. And I also want to thank my collaborators. And also today, this is a good opportunity for me to finally appropriate to show my thank you to Mana, thank you, Gwen, David, Ron, and all the supporting stuff. With all this, now my research would be possible. And also, I guess I never really thanked that in a presentation is the generous startup grant that comes from UH Manoa. So with that, I would like to take any questions.
Any questions for Ray? Ray, I just want to mention that when your paper came out about the bug that you found in the software, I was actually at an NSF meeting for the Advisory Council for Cyberinfrastructure, and it was all the buzz and everyone was talking about it. And I said, wait, that's our researcher running on our resources who made that discovery. So
Yes, exactly.
It was great to be able to say that. And yeah, you had some people running scared, I have to say.
Thank you. I'm glad.
Is Peter Sadowski, and I think you can just take over the screen. Awesome. All right, Peter, take it away.
All right, can you guys hear me? Yes.
Oh, thank you. So I'm Peter Sadowski. I'm in the Information and Computer Sciences Department, and I also work with the Hawaii Data Science Institute and the AI Precision Health Institute at the UH Cancer Center. So one of the great things about living here in Hawaii and doing science at UH Manoa is that we have all this unique access to special data from astronomy, geosciences, oceanography, ecology, and a very diverse population for the health sciences. So Hawaii is really a playground for data science where my research is focused. So my lab specializes in artificial intelligence, and to build artificial intelligence, you need learning from data. And the latest machine learning techniques all make very heavy use of HPC. You need tons of data and you need GPUs. It's basically impossible to do artificial intelligence research these days without GPUs. So Mana is this great resource that makes my research possible. And it's a great research tool for students who can access GPUs for free to try these things out. So a lot of the AI research these days is focused on computer vision, speech recognition, and natural language processing. But my group focuses on some science applications of these same techniques, including medical diagnosis, weather forecasting, and a couple of the things that I'll share with you today. So the key aspect of Mana that we're taking advantage of here is the GPU cluster, which enables us to do lots of matrix multiplications really quickly. So here's one application. It's a collaboration with Masato's lab in biology. We have hours and hours of video, and we want to use computer vision techniques to track the movements of fish from these videos so that we don't have to do it by hand, right? So this is accelerating science by helping with automated data analysis. So these days, this is relatively easy to do with the latest software packages and tools. Another fish project that my students are working on is to help automate fish stock estimation. So we have a giant data set of 1 million hand-labeled images of fish that were taken by fishers in Indonesia. And running it through our AI system, we're able to detect fish from these very noisy images with 93% accuracy. And we can actually classify the species of the fish from a set of 300 different species with 94% accuracy. We can also estimate the size of the fish, which is important for estimating the populations at a large scale. In my collaborations with the UH Cancer Center, we're using the latest computer vision neural network techniques to analyze DEXA data and breast cancer imaging data. So this is a project my student has just submitted this week, predicting mortality from full body X-ray images. So the machine learning algorithm is picking out features of a total body X-ray that indicate that someone is at risk of falls or other aspects that could lead to death. So one of the great things about artificial intelligence is that you can analyze data that humans are bad at looking at. So, traditional computer vision tasks, it's just automating what a human could do. But with artificial intelligence, you can also analyze data that a human doesn't have any intuition about. So a lot of the stuff we're doing is with physics and atmospheric sciences, and you can take these new AI techniques to analyze satellite data or historical weather data. So this is a project in collaboration with Christina in atmospheric sciences, where my undergraduate student, Michael, is predicting cyclone genesis from global reanalysis weather data. And by using explainable AI techniques, he's trying to connect, he's trying to understand what actually causes these cyclones. Another project we're working on is to predict cloud cover over Hawaii. So every time a cloud goes over Honolulu, it causes this sharp increase in energy demand because Honolulu actually has more solar power generation than any other city in the US per capita. So this is a burden on the electric company, which has to ramp up energy production every time there's a cloud. So in this project, we're predicting cloud cover over the city at 10 minutes to an hour. We also have a big NSF grant to analyze data from the new Daniel K. Inouye solar telescope on Maui. So this is taking observations from the DKIS telescope and predicting the structure of the solar atmosphere. So this is pretty interesting, big, high-scale, high-dimensional data. The solar atmosphere has this really complicated physical structure with all these magnetic fields. And as light passes through those magnetic fields, it gets polarized. And the DKIS telescope is able to measure the polarization of the light. So from this huge data cube of information collected by DKIS, we can try to reconstruct the 3D atmosphere of the sun. So the end result of this is going to be faster processing of the telescope observations so that the astronomers can instantly see what's going on in the atmosphere of the sun, rather than waiting 30 minutes to an hour for this slow computation. We also have a collaboration with biologists here on campus, and we have a big NSF grant to use deep learning to analyze microbiome data. As with all these projects, we're really designing new neural network architectures that take advantage of the particular properties of the problem. So my lab really specializes in neural network design principles. In this case, we're taking advantage of the evolutionary information to design a tree-structured neural network that captures the phylogenetic tree structure of the microbes we're analyzing. So this is my group. We have expertise from a lot of different fields, but the general principle of my group is designing AI systems for these science applications. And Mana has really made this possible, and it's been a really fun 4 years since I've gotten here. Thank you.
Great, thank you, Peter. Any questions for Peter about all those cool projects? Any questions? Okay. All right. Thanks, Peter. I think our next speaker is Renee Bellinger. There you are, Renee. I see you. We can't hear you. Got to unmute.
There we are.
Okay, well, welcome. Really glad to see you all the way from UH Hilo, so far away. Thank you for joining us.
All right, so I just want to do a check to ensure that my screen is sharing. Is it set up proper?
Yep.
Okay.
Yeah, we can see your screen. Looks good.
Okay, super. All right, there we go. So good morning, and thank you very much for joining on this Hallmark Day. And I thank the presenters or the organizers here for an opportunity to present research that I conducted as a component of my research over at UH Hilo. I moved to Hawaii in 2015 as a postdoc, and suffice it to say, none of my progress would be possible were it not for the Mana team and the Mana computational resource. Literally, all of my projects have relied upon Mana and the exceptional UH Cyberinfrastructure team that is unparalleled in terms of providing help and support when in a pinch. So uniting interest in this celebration is our appreciation for technological advancements. So first, I'll draw some parallels to the advancements in computational power And then that of how genomics has changed and kind of grown over the past decade, following some of the same trajectory of the advancements that we see in computational, the computational infrastructure. I don't know if you can see this screen share thing, so I'm going to, hopefully it's not there. Okay. So for the last 50 years, Moore's law has been used as a measure of progress in technology. And basically this law states that the numbers of transistors on microchips doubles every 2 years. And at the same time, we've seen a great degree of progress in genetic sequencing technologies that followed some of the same trajectory. Our first sequencing machines came on the market around 1986. However, there was not very much sequence that was produced in each run. And so shown to the left, you can see the speed of sequences as in kilobases per day. And recall that I put a little DNA ladder here so you can see what I mean. So each little ring of this DNA ladder is considered a sequence. And so we've had this absolute tremendous change in how much sequences can be sequenced in a single sequencing run. A major change and a major advancement in genetic sequencing technologies was around 2007 when the first next generation sequencing platform came on the market. And this platform, the Illumina, produces very short reads, so like around 50 base pairs, but millions and millions and millions at a time. And then next on the market came the specific biosystems. Well, there's lots of different things on the market, but the next major advancement was long read sequencers, of which Pacific Biosystems is one. And in this case, the sequences can be tens of thousands or even hundreds of thousands of base pairs long. And at present, our speed of sequencing per day using these third generation sequencing methodologies is over basically a trillion sequences in a single sequencing run in a 24-hour time period. These advancements in genetics have led to multiple types of assembly strategies that have been improved slowly over the years. Now, if you can imagine, if you have short reads, it's kind of difficult to put together a genome, especially when it's extremely complex and has a lot of repetitive elements. And if you consider to the right, if you have these long reads, it's a lot easier to put together the pieces of the puzzle. And as a consequence, over time, our genome assemblies have become a much higher quality in terms of what we're able to produce as an accurate representation of the genome itself. And concomitant to all of these genome changes and in improvements in sequencing technologies, we've seen a rapid increase in numbers of genomes sequenced per year, of which there's around 8,343 animal genomes currently available through the USA's public repository, the NCBI. That was as of yesterday. And for plants, we have just over 2,000 genomes. And it's kind of interesting, I noticed this trend here, and for the plants over the past few years, there's almost like a bit of a stagnation. So there's definitely a bias towards animal and plant genomes, a bias over animals. And so what makes Hawaii extra interesting in terms of genome sequencing projects is Hawaii is very remote. And so when there was a founding event by an animal, or a plant, it was a rather infrequent event, and they encountered these novel habitats and novel ecological communities. And as a result, these animals and plants evolved these extraordinarily diverse forms. And so you might have, for example, one bird species made it all the way over to the Hawaiian Islands, and it diversified into over 70 different distinct forms or distinct species. Another very famous example of these adaptive radiations are the picture-winged Drosophila flies, and then there's a number of different plants. And so today I'll highlight two genome sequencing projects that were only possible because of Mana, and that highlight two endemic species to Hawaii. The first is the alala, the Hawaiian crow, and the second is a plant called bidens. This one is Bidens hawaiiensis, and it's Ko'oko'ulau. So first, the alala, we did a genome project. This one was led by Jolene Sutton, and you can read about it in Sutton et al. 2018. The alala is a crow that is endemic only to Hawaii Island. It's extinct in the wild, and it's one of the rarest birds in the world. We assembled a long-read PacBio genome in order to understand causes and consequences of population decline and to inform breeding programs. And so the first step of this goal, of course, is to produce a genome that serves as a foundation for additional research. And our long-read PacBio genome, I'll walk you through how to read through this graph. So up here on the top row, we have the alala. And this blue, the lighter blue, are all of the single copy genes. And this is a measure of how many genes our genome assembly was able to recover from this genome assembly project. And we recovered over 95% of all of the genes that were expected to be contained in the genome using this benchmarking gene data set. And that data set was comprised of just under 300 genes. That red portion of the graph is the genes that are missing, essentially. And you can see that all of these different bird species, the alala, American crow, hooded crow, zebrafinch, hummingbird, and chicken, are all missing a couple genes. And our alala genome was one of the best ones that had been made, assembled to date, like highest quality for any bird species back in 2018. Now, plants are more difficult in terms of genome sequencing projects because they can have giant genomes. So shown here, you can see the breadth of the genome size in different plant species, over 2,400 fold. Now, lucky for me, the Bidens hawaiiensis genome turned out to be around 7.4 billion bases in its hexaploid form. So both, it has 3 monoploid genomes and then 2 sets. And so basically that was the entirety of the genome size. And we were interested in this plant because the colonization of the Hawaiian Islands was by a single founding event. There's 19 species. It has this tremendous morphological diversity, more so than all the continental members of the genus combined. And so that's why we were interested in it for a genome sequencing project. And shown up here is our Busco representation, where C means the complete genes here. That's the single complete genes. And you see how that purple is proportionally smaller for these single genes than what we saw for the alala. And that's because much of the genome is duplicated because it has that, because it's hexaploid. It has 3 different sets of chromosomes and then 2 from each parent. And the number of missing genes was very low in terms of the overall assembly method. So we got an extraordinarily high quality genome for this plant species that actually has a really big genome relative to all of these other plant species that we compared it against. And these are all in the daisy family, so Asteraceae. And so we kind of benchmarked the state of Asteraceae genomes as of 2022. So this paper just came out. You can read more. It's in Journal of Heredity. And then we also compared our results to these other plants that were polyploid. So you can see those plants also have these high numbers of duplicate genes. And that's kind of like the final frontier in these genome assembly projects, is to figure out how to separate out all of these monoploid genomes from each other. And so the utility is we want to look at evolutionary processes, and there's a lot of species that are endangered, and we can use this information for conservation purposes. And ultimately, we want to understand how genomes are a part of this organismal response to the environment, and Hawaii is a perfect place for that, of course, because we have these complex, novel communities. And so with that, I want to acknowledge collaborators who contributed to this, and a huge shout out to the Mana team for all of the ways in which you support and enable our conservation genetics research, which truly could not happen without you. Thank you very much. I would be happy to take any questions.
Thank you, Renee. That was really interesting. Any questions for Renee about some of her works? Renee, I'm interested in your is it the Hawaiian crow, the alala?
Yeah,
So how were you able to get the samples? I mean, from a
that
reading
was,
program, or how did that
yeah,
work?
it's a great question. So that was in collaboration with the San Diego Zoo Institute for Conservation Research. So we work with Bryce, and then Cynthia Steiner and Oliver Ryder are also a part of that project. In that particular genome sequencing project, PacBio awarded a grant to us basically, and they sequenced the genome for us and provided us the final assembly as kind of a proof in principle for this technology when it very first came on the market for these long read sequencers. So we had a tremendous opportunity to learn from and work with the companies that were producing the actual genome sequencers. And then this research is continuing in that we have samples from the 1800s, from when the population of the alala was much larger. And we're comparing that to a set of contemporary samples that are representative of the individuals that are in the breeding program right now, and then some of the wild birds, their genetic diversity. So examining how genetic diversity has changed over time, essentially. And then hopefully that information can be used in the breeding program.
Very cool. Thank you.
Thank
Any
you.
other questions for Renee? Let's see. All right. Thank you so much for joining us. We will move on now to Samantha for your talk. Welcome.
Hi, thank you. Can you guys see my screen okay? Yeah,
Yes.
okay, thanks. Okay. Good morning. Let me move the, there we go. So good morning. My name is Samantha Streicher, and I'm a postdoc at the University of Hawaiʻi Cancer Center. I'm going to give a short presentation on how I use the Mana High Performance Cluster to perform genome-wide association studies, or GWAS, of body fat in the multi-ethnic cohort. So for some context, obesity is an established risk factor for type 2 diabetes, cardiovascular disease, and 13 different cancer sites. Even stronger associations have been found with body fat location and disease risk. So for example, visceral fat, which is located internally in the abdominal area, shows even stronger associations with type 2 diabetes and breast cancer compared to subcutaneous fat, which is the fat in the abdominal area that we can see, or even BMI. Other body fat locations have been examined with the risk of disease, but it's too early to draw any conclusions. Body fat distribution also differs by race and ethnicity. Japanese Americans tend to have lower amounts of BMI, but they have the most visceral and pancreatic fat, and African Americans tend to have higher BMI, but they have the least visceral and pancreatic fat. Identifying genetic underpinnings of body fat distribution in a multi-ethnic cohort may ultimately contribute to screening tools to identify high-risk individuals for race and ethnic specific fat location and subsequent disease risk. So I use a data set that's housed at the University of Hawaiʻi Cancer Center, the Multi-Ethnic Cohort Adiposity Phenotype Study, or MEC-APS, and they recruited around 1,800 participants between 2013 and 2016 from Hawaii and Los Angeles. They recruited participants from 5 different racial and ethnic groups: African American, European American, Japanese American, Latino, and Native Hawaiian. The MEC-APS participants all provided a blood sample and underwent anthropometric measurements, DXA scan, and abdominal MRI. The genotype information was produced on the MEGA-X array. An array will look at specific variants, and the MEGA-X array reads about 2 million genetic variants. And it has the greatest coverage of genetic variants for any multi-ethnic population. Additional genotypes were imputed, which means that they were predicted based on a reference data set with subjects who have genetic variants for all of their DNA. So this gave us another 7.5 million genetic variants. The imputation was done on the University of Michigan imputation server. The goal of my research is to examine the association between 9.5 million genetic variants and the MRI-assessed adiposity outcomes, such as pancreatic fat or visceral fat, and around 1,800 participants. So on the right here, this is some example data that I work with. It's a VCF file, and you can see it viewed on the Mana high performance cluster, and it has both information on subjects and genetic variants. So the first column here shows the chromosome number for the genetic variant, followed by the position, then the ID, then the allele that's most frequent for the genotype, and then the allele that's least frequent for the genotype. There's a little bit more information about the genetic variant, but I do want to bring your attention to these genetic codes that you see here. So this first code is for one participant in my data set. This is the genetic code for this variant that I was just talking about. This participant also has 9.5 million other genetic codes for all the other 9.5 million variants. There are also another 9.5 million genetic codes for each of the other 1,800 participants in the data set. So I hope this viewing this and my talking about this gives you a sense of how large the data set is that I'm working with. If I were to try to run any of this analysis on my local computer, it would completely crash my local computer. And as I said before, I'm a postdoc, so I don't have a lot of research money. So I use the free nodes on the Mana HPC, and it's really been an asset for my research to be able to do this, to analyze these large data sets. I also use programs that are only Linux-based, so they're compatible with the Mana HPC, but not my local Windows computer. I use programs such as VCF tools and BCF tools to convert files that you see here on the right to other file formats that are compatible with analysis programs that I use, such as Eigensoft and Planck. So I've conducted many GWASs on Mana, but I found the strongest associations with the GWAS of pancreatic fat and the GWAS of visceral fat. So I'm going to briefly go through those GWASs now. So this image is of a Manhattan plot for the GWAS of pancreatic fat. On the x-axis is the chromosome number 1 through 22. And on the y-axis is the negative log 10 p-value for each genetic variant. It's called a Manhattan plot because it looks like the Manhattan skyline with all of the skyscrapers. This red line also demarcates the genome-wide significance threshold, which the p-value is equal to 5 times 10 to the negative 8th, or a negative log 10 p-value is 7.3. So for the GWAS of pancreatic fat, I found 2 genetic variants, one on chromosome 13 right here and one on chromosome 6, that were found to be associated with a decrease in the amount of percent pancreatic fat at the genome-wide significance threshold. The genetic variant on chromosome 13 was most common in European Americans, and the genetic variant on chromosome 6 was most common in African Americans. The genetic variant on chromosome 13 was also associated with an increased amount of the obesity-related biomarker, sex hormone binding globulin, or SHBG, and was associated with a decreased risk of type 2 diabetes. For the GWAS of visceral fat, I conducted the GWAS separately in males and females because visceral fat does differ greatly between males and females. So in males on chromosome 2, I found one significant signal. It was associated with a decrease in the amount of visceral fat at the genome-wide significance threshold. And in females, one genetic variant on chromosome 13 was suggestively associated with an increase in the amount of visceral fat. So suggestively means that it didn't quite make it to the genome-wide significance threshold, but it was very, very close. The signal on chromosome 2 was most common in African Americans, and the genetic variant on chromosome 13 was most common in Japanese Americans. The genetic variant on chromosome 13 was associated with a decrease in the amount of SHBG and possibly associated with an increased risk of breast cancer. So thank you very much for letting me and anyone else who wants to at the University of Hawaiʻi Cancer Center to access Mana. It's really been an asset for my research.
Right, great, thank you. Thank you, Samantha. That was super interesting, wow. Any questions for Samantha? Anyone have any questions? Samantha, I'm curious what you'll do next with this data. This is a really interesting data set and pulling all kinds of interesting things out. What are your next plans?
Well, so actually from the visceral fat GWAS, the variant that we found, as I said, it was associated with visceral fat and most common in Japanese Americans. And we've actually found that it's even more common in the Okinawan population, which we've been able to isolate based on actually using the high performance cluster and Eigensoft and looking at some principal component analysis. And Okinawans are known to also have a lot of longevity, but then they also have a lot of visceral fat. So it's actually gotten me into research to study that population and kind of try to understand what's going on with the possibly like a genetic environmental component to that population.
Great, thank you. Any other questions for Samantha before we move on? All right, well, thank you very much for joining us from the Cancer Center. Really great to have you. So our last speaker today is Karoliina Heyduk, who's going to talk about plant genomics. Oops, sorry, Michelle.
Good morning, and thank you so much for having me today. I'm excited to tell you about what I've been working on here at UH Manoa since I started my faculty position. I'm an assistant professor in the School of Life Sciences. I started in 2020, which has been a delightful time to try to get a lab set up. But one of the highlights has been the ease of getting resources here at UH Manoa on Mana and getting the computational side of my lab set up. So my research focuses on understanding how plants have adapted to live in sort of extreme environments. Here's one picture of such an environment. This is the Mojave Desert in Southern California and Nevada. And lots of desert sort of species live here. They have to deal with extreme lack of water, high temperatures, and high sunlight. And so my work encompasses two main aspects. One is sort of the plant's physiology and understanding how those traits have evolved to allow plants to withstand those environments. And the other is the genetics and the genomics of those traits. So all of the traits that we observe in organisms are encoded by genes in the genome. And so my lab focuses on understanding how these genes evolve and how they contribute to some of the functional diversity that we see in the wild. So again, my research is looking at the plant's genetics, genomics, to understand how that affects trait variation, which in turn allows plants to grow in some of these harsh and unforgiving environments. Renee did a great job of introducing plant genomics for me earlier, but I'll just reiterate this puzzle analogy. So most plant genomes are thousands, millions to billions of base pairs long. And the sequencing technology that I mostly rely on are those short reads. So lots and lots of small puzzle pieces that are on average about 200 base pairs long. And so a lot of the work we do has to do with assembling those short reads into either the entire genome or sort of pieces of it, the gene space. So modern sequencing produces these small snippets of the genome, these 200 base pair reads, and we as researchers need the computational power provided by Mana to really assemble those into something that we can make biological inferences from. So hopefully we end up with this really nice complete puzzle at the end. A lot of my work focuses on the evolution of photosynthetic pathways in plants. So photosynthesis sustains basically all life on Earth, but it has evolved modifications across different plant lineages. So plants photosynthesize by bringing in CO2 molecules into their leaves, and then converting that CO2 into a usable sugar. At the same time, they're letting water out of their leaves back into the atmosphere. If you've ever wilted a plant, it's because you didn't provide it enough water to compensate for the amount of water that it's losing through its leaves. So plants that experience really sort of dry conditions like that desert photo I showed you earlier, this is going to rapidly start to become a problem because they're going to lose so much water that they eventually die. So most plants use a type of photosynthesis called C3 photosynthesis. This is what I'm showing here. But it's really susceptible to water limitation or drought stress. There's an alternative form of photosynthesis that I work on called CAM photosynthesis. And this is where plants actually do this gas exchange process. So bring in CO2 and release water into the atmosphere at night when it's typically cooler, right? So it's analogous to if you went for a jog in the middle of the afternoon versus in the middle of the night, you would probably sweat a lot less at night because the temperatures are cooler. So plants are kind of doing the same thing and minimizing the amount of water they lose to the atmosphere through this alternative form of photosynthesis. And so my research is interested in understanding how this trait has evolved multiple times in multiple lineages and what are the genetic components involved in CAM photosynthesis. And so part of our research is estimating the relationships of the species that we're interested in. We do quite a bit of phylogenomics, and that relies on, again, those short read sequences that we need to assemble and also estimate these phylogenies, which depending on the number of tips shown here on the side, can take hours to days using high performance computing. I also do a lot of assessment of gene expression. So this is again done through sequencing and looking across a number of species. These different sort of categories here are across time. So I do a lot of sampling across a day-night period. And that rapidly adds up. So we have hundreds to thousands of sequencing libraries that we need to analyze across a number of species and across a number of time points and conditions. So both of these sort of tools that we use rely on the generation of sequence data, the assembly of that sequence data, and the subsequent analysis. And all of that is definitely aided by resources available here at UH Mana. I also work on the genomics of some endangered plants. So Joshua trees in the Mojave Desert are not normally on fire, but last summer they experienced record-setting drought and extreme conditions that led to fires that burned large proportions of the Mojave National Preserve. So this is a very threatened species when it comes to climate change. And this is a new national science funded project looking at local adaptation. So in other words, are there some populations that are better able to withstand these increasing challenges of drought stress, high temperatures than other populations? And so we're generating lots and lots of gene expression data. This heat map shows high expression in yellow and low expression in blue. We can compare that across individuals, across different populations, and try to pinpoint what genes are responding to the environment and also what genes are sort of fixed across different populations. And this downstream can help with conservation efforts in Joshua Tree. Another aspect of plant genomics is actually looking at everything except the genes. So this diagram here is showing a number of different plant species. Their genome size is represented by the black bar. And then the proportion of the genome that is composed of repetitive DNA or transposons is represented in red. So transposons are often thought of sort of junk DNA because they don't encode for genes themselves, but they can have a lot of impacts on the plant, how it functions and how it behaves and looks. So you can see in some species, the number of these transposons makes up the vast majority of the genome. And so my research group is also interested in looking how these transposons contribute to trait diversity in different species. And in the species I work on, the genome is 3 billion base pairs, and 80% of that genome is these transposons. So we're talking quite a bit of data represented in this repetitive content. I wanted to end by highlighting the use of Mana in a class I taught last spring. This is BOT 669. This is molecular phylogenetics and evolution. It was a graduate level course where students could use their own data or could download data from available databases to ask questions about evolutionary history of organisms. And so the students all were onboarded thanks to the excellent Mana staff and ran programs themselves and wrote term projects, final project papers that in some cases were nearly publishable results. So this is an excellent use of Mana that I've really enjoyed getting to work with students on the cluster. So I'll just add that I can't emphasize enough how great it has been to work with the Mana staff and get the computational side of my lab set up. it's equitable and it maintains integrity compared to other systems that I've had to use, where suddenly we've been told on other systems that, we have to downsize our amount that we're storing on the cluster, which is not a fair thing to ask after we've already been storing all that on there. And so it's a really robust resource. Additional programs have been really easy to install for our group. I think there's additional opportunity with regard to teaching that I'm excited to continue this coming fall when I teach that graduate level course again. So again, thank you so much for the organizers for inviting me to talk about my use of the HPC system here, and I'd be happy to take any questions.
Excellent. Thank you, Karoliina. Thank you for sharing your experience with your students on the cluster. I know that a number of other folks have also done that, and we're really excited to hear that. And what year are these students? Are these?
They're graduate students, so usually in their first or second year of a PhD or a master's program.
And had they had access or used a supercomputer before
For
this?
the majority of them, no. This is their sort of first instance of really handling data that required an HPC system.
Awesome.
Yeah.
So they feel empowered now
So yeah, hopefully.
take on even more. Okay, well, other questions for Karoliina before we go forward? Any questions? I'm looking around, maybe no. All right, well, this has been really fun. We do have one last thing to show you. We have a little, for those of you who haven't had a chance to visit our beautiful data center, we have a little virtual tour to show you what it's like once you're in there. And if you can, Michelle's going to turn that on for us. Ron, do you want to narrate?
So we're panning over to row G. All of Mana exists in row G, which is just one row in the data center. And we have 357 servers, almost 9,000 cores. Once we added GPUs, we needed to have active rear cooling doors. So we have these kind of expensive, but very effective cooling doors. This is early Mana, I guess you could say, the original Cray, which is down towards the far end. So it's on this side of the building. And these are all the Cray. So the cooling doors allow us to put, instead of just 3 of these enclosures, we can actually fill the thing up. This is the original InfiniBand switch on the old side of Mana. And you can see Mana supports over 90 disciplines, 6 campuses. This is the MRI rack and some condo nodes, and here are GPU nodes from the MRI. So we showed you just a quick view of some of Mana. Thanks for coming. Any questions? It's a lot quieter than it is actually in the.
I thought you were going to play the music from 2001, Space Odyssey.
Question, if I can ask.
Yeah.
Is the space in row G pretty much occupied, or what do you guys see Mana grow in the next, I don't know, decades or something like that?
Well, we have at least 3 cabinets left. It's there are 19 cabinets in the row, and probably, power may be more of an issue, but I mean, it's basically at some point the 2014 nodes will probably get retired, and so as long as the compute power keeps going up, that Mana should be able to at least maintain modern stuff. And we do have some other access to other parts of the data center as well. We have the Jetstream 2 is actually in a row E. So there are other parts of cyber infrastructure around the data center.
Okay, we just need to write more research plan and buy more nodes.
Any questions from our folks, our remote folks for Ron or here's your chance to ask the president a question since he's sitting right here. Well, I'll just add that I think the next frontier for us is really getting good solutions for managing all the data that all you folks are generating. We know that with a lot of the machine learning and artificial intelligence, as well as all those giant genome data sets, it is a challenge for us to figure out how to be able to store your data in a cost-effective way and have you have good access to it. So that's our next big project that we're working on. We're working on a storage solution for that. I think the CC Star Award that Sean led and won will contribute to that. And so it's always helpful for us to hear back from any of the folks that use our cluster to see if it's actually meeting your needs. And we'd love to hear from you. And we also love to hear all your compliments. So thank you all for being so gracious and you're complimenting our staff and all that. That means a lot to all of us. And yeah, so any final comments before we let folks go for the day? Any final words, David?
Oh, just thank you for, this might be the most fun I have all week.
Okay, thanks everybody. Great session, really enjoyed it.